Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Good evening, everyone. Thank you. Welcome to The Hideaway, our host this evening at Breadfruit Plaza, and hello to our viewers at home. We're going to take 3 seconds for everyone to do a little Om. Whoa. Okay, we're going to keep the rain away tonight. Just a reminder, if it rains, the best thing you can do to help us is get out of the way. So if it rains, for the newcomers who weren't here the other night, run to the bar, order a drink, relax, and we'll be back with you in 5 minutes. We have a little rain backup tonight, a backup plan, so for sure it won't rain, we hope. Okay, so let's get started. Thank you to the Lions Club, who help support our live streaming, which makes it possible to watch every presentation after the fact as well as live, and they also go on YouTube in case you didn't know that. This program is made possible through the support of many, but our primary financial supporters are the Culture Fund of the Caribbean, Public Entity Saba, and the Nature Environmental Policy Plan. The program is not possible, though, without many, many sponsors from hotels, jewelry makers, restaurants, other organizations, so please pay homage to all of our sponsors when you see them. Tonight's speaker is generously sponsored by the lovely Jeff. Hi, Jeff. At Althea's Cottage, which is right up the road, beautiful place, and he's a wonderful sponsor of all of our programs. For our newcomers tonight, we have a great raffle going on this year. As always, $8,000 worth of prizes, $5 a ticket, announced at final night on Monday, but you don't have to be present to win. And for our streaming people, you can also contact us to buy tickets, and we'll make sure they get there. Upcoming events. Saturday morning at 8 o'clock is the hike with tonight's speaker. He's going to tell you more about why you should join that cool field project and get up Saturday morning and get your blood flowing. Our next speaker will be at Calibre Cafe Saturday at 5:30, having a little technical difficulty with our slides. And that will be with Aaron Pilnick. Give us a wave there, Aaron. And you can sign up at the tent for any of the field projects that are left, and there's no reason you don't have to sign up for the presentations. Just come and support our sponsors. Any field project that you participate in, you get a free, wonderful hat, sustainably made with our cool logo on it, so don't forget to ask for it. And let's get going with tonight's speaker. Hold on to your spores. Dr. David Schmale is landing on Saba. A professor, drone pilot, and microbial storm chaser, David tracks airborne microbes that travel across continents and oceans, impacting agriculture, ecosystems, and even human health. As director of the Translational Plant Sciences Center at Virginia Tech, he blends cutting-edge tech with wild biological questions, whether it's flying drones into clouds or wrangling slime molds with students. Named one of Popular Science's Brilliant 10, David brings big science with an even bigger heart for education. With a passion for storytelling, outreach, and microbial mysteries of the sky, this is one scientist who truly knows how to make science airborne and awesome. Let's give a warm Saba welcome and a round of applause for Dr. David Schmale.
Thank you, Lynn. I'll take that clicker from you. Fantastic. On Monday, I asked Jeff, hey, Jeff, what's the chances of me being able to actually see through the clouds on top of Mount Scenery? He says, I don't know. Yesterday it was really good. You could see the tower. So I decided, you know what? I'm going to give it a go. So all 1,064 steps, if I'm remembering correctly. And so that's how I started my journey on Monday, of course, here on Saba. With the first stop, of course, at Mascajones, a little panorama. And, of course, you can see still clouds there. But of course, I was still hopeful that once I got to the top of Mount Scenery, I was going to see something. Of course, along the trail, I spotted, of course, the 3 different shelters. And the shelters are really nice because they tell you how many steps you have left to go. And just when you're like, oh, yeah, I'm like halfway there. No, you got another 600. And so it was sort of neat markers along the trail. And, of course, certain parts of the trail were really cool with what appeared to be some old rocks, maybe from some terraces that were on the hill from many years ago. Of course, I also had the opportunity to run into some wildlife. There was a pair of racers, which interested me. At first, I thought it was just one. I got a little bit closer, and I might have actually interrupted a little snake love during that moment. And then, of course, the higher I got up to the mountain, I ran into the elusive high-altitude chicken that was also on the trail, actually near the top. So both native and, of course, introduced species. Of course, making it to the top was super cool. And on my way up to that last little bit after I'd passed the tower, I ran into a guy who was coming down. I said, oh, did you get a chance to have the view in the clouds? And he says, well, a little bit. I said, okay, well, about how long do you need to wait? He said, well, at least about 30 minutes to give yourself enough time to see it. So got up there, and of course, I was in the clouds. And then I just waited, went up to the rock, and of course, moments later, the clouds started parting, and I got just this really incredible view, which was fantastic for me. And of course, as soon as I was able to enjoy the view, it disappeared once again. And of course, the airport down there on the left, for those of you that have made it up to the top, it's just a beautiful spot. And of course, as soon as I pan back around, it's all enshrouded in clouds again. And so, given that my talk focuses, of course, on the atmosphere and how we're all connected to the atmosphere, this is going to be a big theme. I want to sort of come back to the storytelling aspect that Lynn introduced as part of my introduction. The atmosphere is actually carrying stories from distant lands. And so up there, as that wind was blowing, and it was blowing pretty fiercely, and those clouds were rolling in, that atmosphere was from near and far. And we'll talk about some of those stories today. So I started my journey here with outreach, actually sharing, of course, some information about these wonderful flying seeds that native trees you have here on the island, West Indian mahoganies, produce. And so there's a beautiful specimen down here by the museum, if you haven't checked it out. They, of course, produce these fruits, and when these fruits dry, they actually shed these wonderful flying seeds. Now, these seeds spin like little helicopters. They have a formal name. They're called samaras. And I was fortunate that I asked Lynn and Emily if they could actually find some many months ago when, of course, the trees were shedding them there on the island. And it's my understanding that with walks with the dog, among other things, they collected quite a few. And so I have some here, if you haven't seen these seeds before. I'm going to demonstrate exactly how one of them flies, and let's hope it actually spins for y'all. And so, oops, actually I just broke the end of it. Just trying to get it out. I've got four attempts, right? So the seed itself is, of course, at the bottom of the wing. And then as the seed flies, it spins. Do you see that? It's pretty cool, right? We'll do it again. See how it spins? And of course, with a little bit of wind, that spinning seed can then be carried over a considerable distance. And of course, the bigger the tree and the higher the fruit, the higher the seed. And so many of these seeds can travel considerable distances. And so I'm going to actually pass these around. I ask that maybe you just don't reach in there and try to break them, but you can take a look at the seeds inside the bag. And I was inspired by Don's presentation when he was passing around those really cool sponges. He was letting everybody squeeze those. I'm not letting you squeeze the seeds, okay? All right? So, of course, a big part of this, this whole see and learn concept is teaching a bit about the island, but a bit about, of course, the other type of work that we do. And now, for some reason, something is frozen. Oh, did you toggle something? My slide's changing on here, but it's not changing on the screen. Yeah, I got it on the screen. It's just not changing on the screen. It's not my clicker because I can see it moving on the Mac. Well, you know, Don and I were joking. He had some challenges the other night with the clicker. And so I'm like, okay, I got my clicker. I got fresh batteries. I've got this problem solved. And now we encountered another one. So, you know, one of the themes that we're all talking about now is resilience. This is where we're at tonight, right? Well, you know, some people. Oh, now I've lost my mic too. Some people say that I'm an airhead. Yeah, thank you. Well, we were going good. Oh, my voice is back. I was going to say just good until the flying seeds took flight. So I'll give you a little background as he's getting everything back up. In sort of developing the outreach lessons, I came up with this idea of actually showing the students these flying seeds and then asking them to make their own flying seeds, following a little template. And so actually, while we're waiting for him to bring it back up, I'll show you what that looks like, and then I'll show you what the students did. And so with, of course, a little piece of paper, some patience, and cutting some straight lines. Hey, there we go. We're back up, I think. Yes, we are. Awesome. Okay, well, I'm going to keep with my story here. And a little paper clip and a little bit of folding, you can, of course, make your own flying Samara. And so take a look at this. Look at that. Pretty cool, right? And so we did this with grade 3 students. Now, you might notice that my voice sounds a little hoarse. That's because I lost it while working with the students. Beth was with me. She was wonderful, and controlled chaos might best describe our interactions. And so the students here are on Wells Bay Road, and I'm going to show this video, and you'll see them. I've asked them to, of course, count down 3, 2, 1, throw them all in the air.
2, 1, toss! Woo! All right.
And so they were pretty excited. This is, of course, a photo, and certainly the excitement on their faces really demonstrated this wonderful opportunity for all of them, and also that, you know, flight connects life. Again, connection is a big part of this, and that wind really connects people, places, and, of course, the atmosphere as a whole, and plants and other things that we're going to talk about today. So the title, When the Sky Comes to Visit, is something that, of course, we're hoping the rain doesn't come to visit tonight. I'm going to be talking about hidden highways, and in doing so, I'm going to be talking about some wonderful work that really the credit goes to the members of my lab and our sponsors. And so I want to make sure to acknowledge them. We've had the great fortune of having uninterrupted funding from the National Science Foundation for more than 2 decades, support through the United States Department of Agriculture. I had the wonderful opportunity of having a Fulbright in New Zealand with my family. We were there actually for a year and a half during the pandemic. If you'd like to talk about New Zealand, I'm going to show some research and footage from New Zealand. It's a very important place in the world, and of course another wonderful island nation. Support from NASA in studying some of our African dust work, internal support at Virginia Tech, our center, and then of course now we get to add See and Learn. So thank you, Lynn, Emily, and everyone else there. So I'm going to talk about connections, but I want you to think about interfaces, and interfaces are where different systems come together. This is a dandelion flower, and you're going to see in slow motion wind sweeping across the dandelion flower. Now, for anyone in the U.S. trying to maintain a nice little lawn, this weed is very problematic. And this is part of the reason why. The wind sweeps across the surface, and these beautiful seeds, which look like little parachutes, of course, get launched into the air, and of course they can travel quite some distance. And so they're not only producing a ton of seeds, but of course their ability to move quite quickly through the atmosphere is advantageous for their dispersal. Now, air-water interfaces are also very interesting, and I'm going to be talking a little bit about those towards the end. This is a beautiful kingfisher bird, and it of course is leaving the air. It's actually diving through the water, and it's grabbing some prey. Now this is probably the easiest part, but it now has to turn around and get back up out of the water and back into the air, and that's a heck of a lot more difficult. And so I'll be talking about processes of aerosolization by which organisms can get out of some system and ultimately into the air, whether it's off of a plant surface or even out of the water. And of course, these interfaces are where these different systems come together and interact, and this is where the connections happen. The mic is going in, I think. Are you still hearing me? Yes? Okay. Could just be my voice dropping down. Now, the atmosphere is actually teeming with invisible life. So if we look up at the clouds now, behind the mountain or just up above, there are microorganisms trucking through the atmosphere. There are particles. There's pollen grains. And in fact, the sampling activity that we'll be doing on Saturday, I brought an air sampler with me and a little digital microscope. So we're going to collect samples of the air right here on Saba and actually take a look at what we actually collected. And I anticipate finding all of these types of particles in the air samples that we collect, and they may vary depending on where we sample on the island. Now, the COVID pandemic, of course, allowed the entire world to really understand the term aerosol and the fact that a virus could be transmitted through the air, could be passed through the air from one human to another, from one host to another. Of course, plants can also be, plant diseases and plant pathogens can also be transmitted through the atmosphere. And so on the left here, we have Asian soybean rust on soybeans. This is an invasive plant pathogen, and it came to the United States via Hurricane Ivan back in 2004. Now, it's my understanding that Hurricane Ivan passed over Saba, but it was south of Saba during the time, for those of you that were here, and it wasn't, of course, as big of an impact as Irma and Maria for the island. Nonetheless, this is the trace of the hurricane, and as it came along, it actually intercepted spores of the fungus that were here in the northern part of South America, and then carried it into the United States. And in a single event, in a single storm, in just one night, essentially, this devastating plant pathogen crossed borders into the United States. And I just wanted to illustrate, this is an actual track of Hurricane Ivan. You can see it, of course, passing over the tip of South America, picking up those spores, and then broadly distributing them across the southeastern United States. Now, the hurricane made landfall on the 16th of September. 7 weeks later, the first report of soybean rust actually showed up in a field in Baton Rouge, Louisiana. And so for 7 weeks, this fungus was basically replicating and infecting plants before we actually detected it. And so early detection becomes an important part of all of this. Now, all of you should be familiar with Saharan dust, because Saharan dust clouds come here to Saba. And in fact, this satellite image illustrates just that. These dust plumes coming off the Sahara, across the ocean, and then here at this point in time, intercepting, of course, this big plume Puerto Rico, and eventually heading to the United States. And I show this image because this is a photo here. This is from July 18th, 2024, from Puerto Rico. And you can see the sky has this orange color to it. And that orange coloration is indicative of those dust particles in the atmosphere. Now, microorganisms actually ride along with those dust particles. In some cases, they're attached to them. And so these big plumes can actually be foundational events for pathogens and other things to move in to other regions. And so the connectedness, the fact that here in Ceiba, the air at certain times of the year, particularly June and July, is connected with the Sahara. So think about that. Now, of course, Virginia, where I hail from, we've got a huge problem, a huge problem in the eastern United States, and that is this invasive spotted lanternfly. Just last month, there were reports of this huge swarm of these insects that were actually traveling near Washington, D.C., and they actually came up on radar. And so this radar picked up this huge swarm, which is in green here on the left, and I'm not supposed to use my pointer, so I'm going to go over here and show you. This showed up under a radar that would usually monitor snow and rain, but there was no snow and rain. Now, interestingly enough, I was in Washington, D.C. 2 weeks later after this event at one of our new campuses, the Innovation Campus in Arlington, and there were literally hundreds of carcasses of the spotted lanternfly scattered all around the ground, literally crunching underneath my feet as I was walking into the building. And so this is a huge issue for us right now. This organism produces these huge egg masses. It reproduces very well, and of course has these huge clusters when it's feeding and laying eggs. Studying life in the atmosphere is, of course, very complicated and requires teams of biologists and engineers. I'm an aerobiologist by training. I study life in the atmosphere, but I can't do that alone. And so I've had the great fortune of working with my wonderful colleague here, Dr. Shane Ross, in aerospace and ocean engineering. And he and I have worked together for almost 2 decades, 20 years, studying the flow of life in the atmosphere. We had an annual review where we sort of systematically broke down transport in the atmosphere as basically scales. And the annual review talks about these highways in the sky, how plant pathogens can actually start in a source, an area such as a leaf, an infested plant, move up into the air, encounter, of course, all sorts of challenges from UV light to rainfall, and then eventually be deposited, hopefully, on a susceptible host. And these scales of transport, of course, move to many different systems, and I'm going to be talking about some of those today. As we think about these scales, we systematically have defined them as the pathogen scale, where we look at sort of the spore-producing structures, the farm scale, when we're thinking about growing food, into the regional scale. For the United States, it's something like a state, with our own state governments and state borders, and then, of course, the continental scale, where we're moving really long distances. And so I'm going to talk about examples of the work that we've done in all of these different spaces. And I want you to think about a journey, and this is a clip I generated using Sora, which is a really incredible AI tool developed by OpenAI, where you can turn words into videos. And this video will show you this journey that we're going to illustrate today. From the top of a leaf, that spore becomes aloft and then moves along these highways in the sky. Okay? And if you want to have any conversations about AI tools and how you can do this for free, let me know. It's super easy, and you can generate something like this in about 15 seconds. Pathogen scale, let's start here. Spore-producing structures of a fungus, the scale is really very small in millimeters. And I want to introduce an important plant pathogen. This is rust that occurs on leaves of wheat, and the rust fungus produces these blisters. These blisters are called pustules, and each pustule, when it rips open, can produce hundreds of spores in just a single blister. With my colleagues, we've been trying to understand the pathogen scale transport of this fungus from leaf plants. Sunny Young over here has a high-speed video camera. This video camera can record 1 million frames in just a single second, and you're going to get to see some of this video here shortly. So if you use your slow-mo on your iPhone, usually you can record about 200 frames or something like that in a single second. And so this is pretty cool. And then Jonathan Barreco here is looking at a droplet generator where we can target a little raindrop. It can fly down and basically hit this leaf, and then we can watch what happens. And so you're going to see that. You're going to see a raindrop falling down. It's going to impact this leaf surface, and what you're going to see are rust spores getting thrown up into the air. You're going to see that some of these spores actually are lodged on the surfaces of the droplets. Hopefully you can see that in the video. So the spores are actually on the surface of the droplets here. What you can also see are these huge swirls of spores that are being dispersed in dry air right up here as well into the corner. And this was actually a surprising discovery. Of course, we might expect that spores impacting droplets impacting a surface would, of course, engender spores in the droplets. But we were surprised when we actually observed and we tracked each of these individual spores that they were traveling in these microscopic tornadoes. And so, and this was a dry dispersal event that allowed them to move up into the atmosphere. And you can see these little teeny microscopic tornadoes that took these spores up into the air. Now, some of you are holding a cold drink tonight, okay? And if you are, I want you to look up and notice the condensation droplets that are on the outside of your drink, okay? I have a lot on this bottle of Powerade right here. Now, as condensation droplets actually form, if they collide, the energy of the collision causes some of them to jump spontaneously. So you can't see it yourself, but it's happening, and I'm going to show you that it happens actually on lead surfaces. When these droplets collide, they can take spores and other things and move them into the air. And so in this video, you're going to see this phenomenon actually happening. And this is with the former graduate student, Dr. Nath, observing this phenomena on leaf surfaces. So we're looking off to the side. These droplets are condensation droplets that are colliding and then spontaneously jumping away from the surface. And so we've created an analogy that the plant is actually sneezing. It's sneezing and trying to expel the spores. And sometimes it's iterative, so you get one that jumps, it lands, and then it jumps even higher, and then it lands again. And of course, these are really challenging videos to get because they have to be in the same frame. Now, this is the money shot where we wanted to actually see that these spores were actually moving with the droplets. And so keep an eye over here. You're going to see a droplet shoot right at you. Boom! That was really cool. We're going to zoom in and you're going to see it again. There are actually 3 spores sitting there on the surface. The collision of the condensation droplets come together, and then the plant sneezes and the spores basically move away from the surface. Watch this. Boom! And so we needed that to be able to, of course, show that this was really happening. In the laboratory, we made a setup where we could create condensation on a super cooled stage. We put a little fan on there, and then we had these color changing papers where the droplets would land. And then we could go and look at the color changing papers, and we could count the number of spores that traveled with that distance. We had a really nice paper in PNAS describing this phenomenon, which was really exciting for us. Now, that's the pathogen scale. What about the farm scale? Well, one of the things that plant pathologists like to do is we like to make plants sick. And so if you want to understand how an organism is traveling over long distances, one of the things you can do is you can grow a healthy crop, you can kill it, and then you can find out where the pathogen moves through the air. And that's exactly what we do. We happen to be really good at this, actually. And so here I'm going to tell you about a fungus that also affects wheat, Fusarium graminearum. It causes a disease called Fusarium head blight, and this fungus is also producing toxins that are accumulating in the grain. Half of my research program actually studies and quantifies these toxins in grain. We run one of the 3 diagnostic testing labs for this mycotoxin in the United States. This fungus produces airborne spores. They move through the air, and then they can infect not only the local field, but farms in nearby regions. And so former graduate student Dr. Prussian intentionally went out and released the fungus into this healthy wheat field. And then, of course, the fungus produced its spores. And then we tracked the movement of this fungus using DNA-based technologies over different distances all the way up to 1 kilometer away from the source by trapping it on little dishes and then also measuring its movement within the field. And of course, this allowed us to monitor how this pathogen was actually being released and how plumes of this pathogen moved over greater distances from that source field. And so you can see this is really complicated. It depends on environmental factors, and of course the pathogen isn't always moving in one certain direction. It depends on the wind and the connections with the wind and other factors. Now, we've also done some work with the movement of transgenic pollen. And so, of course, genetically modified organisms are often concerning for many people. And so if you're growing a crop that's been genetically engineered and you're adjacent to somebody that's trying to produce an organic crop, for example, you have to worry a little bit about potential contamination. And so we intentionally got a permitted plot in the state of Tennessee in a clear-cut area in some beautiful Appalachian forest. and we released transgenic switchgrass, and we actually studied their transport. There's a little bit of sound here if you want to turn it up just a little bit. And if we don't have the sound, maybe we lost the sound. Well, anyway, there was sound, but this is our field operations for our group out at that site. We bring a lot of people out to conduct these intensive campaigns. We have ground-based sensors that are used to track the pollen, both inside the field and outside the field, a mobile lab, and then my laboratory really specializes in drone-based sensors, and you'll see some of those in action here shortly. Different types of sensors that are mounted on the drone that enable us to monitor, of course, the agent at higher altitudes. And this just shows you sort of how our equipment is situated out in the field, the unique topography, mountain topography of the area, which was a requirement for the experiments, and then, of course, the crew that has to line up for these different sort of collections that we have throughout the day. And we do these repeatedly as the crop, the switchgrass, is releasing the pollen. We do a lot of 3D printing in the lab. Some of you are probably familiar with this technology that enables us to share devices with the world. Anybody with a 3D printer can go ahead and make scientific equipment and then deploy it, doing their own citizen science. And then you'll see shortly one of our drone-based platforms that we use to actually trap some of these agents in the air. This is it right here. The big tower that's sitting above this large quadcopter has a meteorological package, and it has a little spore vacuum. In this case, it's being used to trap pollen, and the pollen gets sucked into this little bubbling fluid that you can see on top of the drone. As air gets bubbled through, it gets trapped in the liquid, and then, of course, you can collect these agents at different distances, both above the source field and downwind. And then we always like to show the good landings. The unplanned landings are the ones that we don't like to show, and we don't refer to them as crashes. They're always unplanned landings. And this is a rather large drone. It's quite a big platform. Now, at Virginia Tech, we have the Kentland Research Farm, which is an incredible facility. This facility has an airstrip for our larger fixed-wing aircraft and our big research bay so that we can do unique experiments. And we've been doing a lot of work with colored smoke. These colored smoke packages you'll see here shortly are wonderful visual tracers. We can't see the pollen moving in the air. We can't see pathogen spores, but we can see colored smoke, and we can buy these little colored smoke emitters. As it turns out, this company has marketed them mostly for gender reveal parties when somebody's pregnant and they want to have a gender reveal party, so they have colors that are sort of matched to that. But you can get a variety of different colors. They're very inexpensive. And of course, then you can train your robots and your sensors to be able to detect and track these plumes. That, of course, was a yellow one. Now you can see a blue one here. And then you'll see multiple colors that we release also at the same time. And this allows us to, of course, validate our transport models, but we can also put these into our research fields, and then we can actually see essentially where the agent is going. This is releasing sort of at multiple heights, and now the drone package can be used, particularly with these optical particle counters that are making near real-time measurements of particle size distributions in the air as the drone can sort of traverse these different plumes. And here you can see it here is we kind of did a lot of rainbow colors out there, releasing different things. Different colors have different signatures, so we can track them with different sources. And of course, when you have really high winds, you can see a plume that's very shallow, and that makes sense, right? A really strong wind, it's going to be really low, but if you don't have any wind, you get this vertical plume. And so understanding this type of distribution is really important if you want to understand where an agent is going. Now, for us in our switchgrass, this was a year later. We went back and we deployed these emitters right over our source, and you can see how dynamic it is. The wind was blowing at one point in time, and of course the pollen that's moving off of there was going this way, but then all of a sudden the wind changed, and now it's going this way. So it's complicated. It's not something that's inherently that easy to, of course, sample, particularly if you want to move a sensor around. And this is just an overhead view of that same plume, first going this way, now changing direction and going this way. And then, of course, since we can visualize the smoke, we can move our sensors into location to then be able to track what's coming from the field. And so that's just illustrated here. Moreover, we're moving into these new AI-based technologies, and one of them is called Detectron by Meta. And so this is an entirely AI-based imaging system that can take these plumes, it can image them. I don't know why it's not running now, but you got the sense of it. Let me go back and I'll run it. Oh, we'll run it one more time. There it is. As the plume's moving around, it's actually automatically detecting the concentration and then mapping it within the domain, which is really cool. Okay, we're moving on now to the continental scale. And I wanted to take a turn back in time. Some of you might remember hearing about these Australian bushfires, which were particularly devastating in early 2020, late 2019, early 2020. We actually landed in New Zealand on January 1st, 2020. And shortly after landing in Christchurch, the next day we actually took our van that we had just purchased, and we drove it out to Akaroa, this beautiful area. And the entire sky was just absolutely covered in ash from these Australian bushfires. And the kids' eyes were hurting. I have two girls. My wife's eyes were hurting. And we were standing on this point looking out. And of course, it was very, very hazy. And this, of course, was wildfire smoke that was coming in from Australia. And it was a major incursion event to New Zealand. Well, our colleague, Dr. Pretorius at Scion, actually took a look at some of these satellite images during these major events that occurred with the transport of the smoke from these Australian bushfires actually moving into New Zealand. And you can see the satellite images on the top. Well, she used a rather sophisticated modeling technology to be able to determine what we're calling air bridges. And these are bridges that basically connect Australia to New Zealand. As it turns out, these air bridges connect a lot of land masses with other parts of the world. It's really fascinating. And of course, you can model these connections at different altitudes in the atmosphere, as you can see here, and then at different times of the year. And so thanks to Ilsa's work, and of course our ability to be able to predict these air bridges, they were able to land a nearly $11 million project funded by the New Zealand government. And we're fortunate, my colleague Shane Ross and I are a small part of this, trying to basically understand these air bridges and understand when incursions or events that bring a pest or a pathogen from Australia into New Zealand can be predicted, so then they can go and be aware when that might show up. And this is Toni Withers, who's an entomologist. She's actually, of course, smiling as she's attracting and collecting some insects in the field. And I had the great fortune of heading back to New Zealand about 1.5 years ago. This is me in front of NIWA and, of course, the Scion team for this project, all getting together to launch it. This is very deeply ingrained in the Maori community in New Zealand, which are the indigenous people of mainland New Zealand. And so New Zealand really has developed this bottom-up approach where communities really drive the research instead of the research driving down to the communities. And it's something I think we can all learn a lot about as we talk more about connections. Now, Don was telling me a little bit about some of his work with fluorescein, and for those of you that spent some time with him looking at dyes and sponges and other things, I was inspired to share some of our work in water. And water also transports agents across the planet. And so in the last 5 or so years, we've moved into the water. There's been quite a bit of opportunity for us to think about transport, but in water systems. And so I'm going to share some of that work here. It's going to start with this research vessel, Connecticut. We had a cruise out in the Atlantic Ocean, south of Martha's Vineyard, where we released a dye, a rhodamine dye, into the ocean, and then we tracked it for many days as it moved from the source. And this was to simulate an oil spill, and then so that we could use some sophisticated tools to find out where it might go. And so you're going to see that here. I'm actually flying the drone. I'm wearing that little yellow jersey there on the front. And we got music for some reason we didn't have before, but now we got it. That's great. The captain of the ship actually was trying to do a spiral, but he didn't account for the actual currents until I showed him the drone images. Anyway, there's a rib that's going out with some sensors to image the dye. I'm flying the drone here, capturing images of the plume, and then you'll see shortly we have a drone-based water sampling platform, which is kind of like a little fishing line with a water catcher on the bottom. We can run out, we can take a picture, we can grab a sample, we bring it back, we swap it out, we go out and we collect another one. And that's how you catch a drone out in the middle of the ocean. We also have underwater robots. These are ROVs equipped with sensors that allow us to monitor plumes of the dye from the surface down. And we've done quite a bit of work with tethered systems. This is Big Bird tethered to our underwater robot. And we have a beautiful little quarry that we can do some of this work actually very close to campus. It's privately owned. This is Regina wonderful research associate in the lab deploying this. And of course, you can see these dyes. This is fluorescein dye, similar to what Don was working with. These robots can detect the dye in real time as we traverse these plumes. And these are great ways for us to actually train our robots, particularly as we move into natural environments. We've done a lot of this in the framework of search and rescue, tracking dummies that of course could be the result of a sort of a capsize event. And then much of our work in the last 5 years has moved into harmful algal blooms, tracking the movement of toxic cyanobacteria as they leave the water and move into the atmosphere, deploying our drone-based systems and our robots to move out and characterize these plumes of cyanobacteria in lakes. This is Grand Lake St. Mary's, one of the most toxic lakes in the United States. It's only about 2 meters deep. It's also the water source for about 3,000 people. And so the person who runs the water treatment plant, of course, is a very important person in the town of Salina. This is Lake Erie. Lake Erie in this part of Toledo actually provides water for a number of people. We've done some work in Austria in some just horrible locations like this one. Gusau, which is of course a glacial valley, sampling water for ice nucleating microorganisms that allow water to freeze at higher temperatures. And of course that's the Dachstein Glacier in the back there. And then this is a campaign we had out in Chatham Island in New Zealand, which was just an incredible location to do field work during the pandemic with the Lakes 380 team. These are the Necklace Lakes, which are these incredible dune lakes, of course, nestled right on this narrow body of land with the ocean on both sides and entirely rain-fed. So, as we come to the end of this conversation, and I don't even know where I'm at on time, but this is coming to the end. One of the big take-home messages for all of you is I'd like you to spend some time looking where systems touch. Life moves in patterns that we're really only beginning to see. We need technologies like high-speed videography. We need robots. We need satellites. We need people working together across different domains so that we can be able to study these processes. Observation is the first step towards stewardship. If we're going to take care of this planet and take care of each other, we need to make sure we understand what's going on. We need science. We need data. Connection really, I'd like to argue, is the story of our planet. We're connected to this planet, and our systems are connected to one another. And the sky and the water, guess what? They're in a constant conversation. So I want you to think about that, and I think we need to listen just a little bit more. So with that, I'd like to thank you for your attention. My voice somehow miraculously held up, I think with the aid of the microphone. And I'll just give a final clip as I was enjoying my breakfast over at the Tropic Cafe there this morning. The clouds just moving across, and you could see the tower emerge just there this morning. So with that, thank you very much. Appreciate the opportunity to be here. Yes?
Are you doing much research because of the wildfires in Canada coming down through the States?
Yes. Yeah, it's a great question. So the question was, are you doing any work related to the California wildfire, or not the California, the Canadian wildfires, and as they move into the States, and of course California has wildfires too that can blow across the U.S. as well. We aren't directly, but we have a large team that we put together to model and sample what we call atmospheric incursions, and to understand these air bridges, and then to establish a sampling network basically around the borders of the United States to understand and predict when these agents might actually come in. And it's always complicated when you're studying phenomena that happen outside of your country for a lot of logistical regions. And so just keep that in mind. So it's a great question.
Has any research been done as to what's been taken out?
Yeah, that's a great question. So she also asked, any research being done with the California wildfires and then the movement into the ocean? And the short answer is yes. There was an incredible report that I read a couple weeks ago about these devastating impacts of the ash fallout on marine communities along the west coast of the United States. And so that's certainly not my specialty in understanding those impacts, but certainly the African dust events in the Atlantic have been associated with devastating CFAM populations and potential causal agents of disease, such as a disease of CFAMs called aspergillosis. Don and I were talking a little bit about this. He's really the disease ecology guy in the water. Great questions. Yep, Ken.
David, fascinating. You just mentioned part of my question is about marine organism pathogens and African dust. How about human pathogens and African dust?
Yeah, that's an interesting question. So Ken's question is, what about human pathogens moving in these dust events or in the air? There was an interesting study done in Japan that reported using a full-scale aircraft flying over Japan that human pathogens were aloft at very high altitudes. And so I think we're starting to get more and more compelling evidence that it doesn't really matter who your host is. And if you produce enough of the agent, enough spores, enough material, there's still a pretty good chance that you can remain viable for a long-distance transport event. So if that little spore is shielded by, a dust particle or a smoke particle, a wonderful colleague, Lita Kobzia in Idaho, has created this field of pyroaerobiology. She's studying how things move with wildfire smoke. And so, you have a devastating pathogen. Oftentimes the control measure is you burn the forest. Well, you might eliminate the issue there, but you can actually create a huge plume for the agent to then move from one location to another. And so I think we certainly need a lot more research in this space, but we also need to understand more about the sources. And of course, in dealing with human pathogens, it's very hard to conceive experiments where you might want to infect a bunch of people intentionally and then understand where stuff goes. So oftentimes it's sort of a pass cast. We know a bunch of people got sick, and then we find out where they got sick from. There's been a lot of work in the red tide domain in sort of respiratory illnesses and mapping those and associating them with this dinoflagellate, Carina brevis, and these brevetoxins would get aerosolized and people get really sick. So those sorts of issues allow us to move into that realm of understanding those events. Good question, Ken. Yes?
Are you seeing differences with the way the atmospheric pressures or movements are happening? Because, for example, like with the African dust, growing up, I don't recall it as a young child seeing it. Whereas now we know in the summer it's totally hazed out. And do you think that climate change or something has an impact?
Yeah, that's a really great question. So the question was, she shared this wonderful story. She doesn't recall in her youth noticing African dust events like she's noticed more recently. And the question was, are things changing? Is climate change part of all of this? It's a tough question because we know with extreme weather events in particular that many things can move very quickly and sometimes unexpectedly. Hurricanes are a great example of that. We are seeing these large magnitude African dust events, and they're occurring pretty regularly in June and July. And they're intercepting regularity the eastern coast of the United States. On the west coast of the United States, we see Asian dust. It comes over a little bit earlier, March, April. And so we intercept that sort of on the west coast in tall mountains out there. Researchers now are using AI models to actually connect the air, the ocean, land, and start to understand the entire Earth as a system. As we begin to train these models on historical data, I think we're going to have a better idea of what the future might hold. And we're not too far away. It's moving so fast. There's a lot of publications coming out in this space where these AI models are starting to tell us, have better abilities to forecast hurricanes, for example, or major African dust events. We need more data. We need more time to be able to figure these things out. And AI is going to help bring a lot of those things together. Great questions. Yep.
How big is the field of aerobiology? And how old is it?
How big is the field of aerobiology? It's a lot bigger since the pandemic. Let's just think about it that way. It would have been an easier question to answer 5 years ago. And of course, viruses don't belong in the tree of life, but they do connect living organisms to, of course, diseases. Yeah, so that's going to be my short answer. I will share that Charles Darwin actually was among the first aerobiologists. During one of his research cruises, he actually collected African dust swept it up and put it into a small little vial, and then that vial was deposited in the museum. And Dale Griffin, who used to work for the USGS, actually did some work with some of those samples. Pretty cool. He's also collected African dust samples on ships out in the middle of the Atlantic. Yes?
Dale Griffin, who was supposed to be here last year and couldn't make it because of storms and recommended you, he told us that all of our clay on Saba is from African dust. Can you kind of answer that?
I can't
without
answer that.
doing
So
a full
Lynn made a
presentation?
statement. I could text Dale and ask him. Lynn made a statement that suggested that Dale attributed all of the clay on Saba from African dust. I don't know if that's the case, and I don't know about the clay here, but certainly dust from Africa does indeed come to Saba. It's probably been doing that for quite some time, and so there's probably that record. But I can't. I'm not a soil scientist by any means. It's a great point though. Yes.
Is there a problem of spewing out a pathogen like you had in the field up there?
Yeah.
Because it could go where you don't want it to go?
Yeah, it's a great question. So she said, "Hey, is there a problem with intentionally releasing a pathogen that could go anywhere?" And the answer is yes. And so this is where communication and connection come into play. With our release recapture experiments, we do them on a research farm. We tend to delay them after the actual season, so we're not impacting growers in the area. And then we try to set up the experiments where they're not going to be in areas where the crop grows. So for example, with the pollen experiment, this was in the middle of the forest, no switchgrass around. And so communication is a big part of it. We also maintain diseased plants in greenhouses, and in adjacent greenhouses, there are healthy plants. And so the best thing you can do is just educate people and let the other scientists know what's going on so they don't get upset and come after you. And so pretty much with everything in life, it's all about communication and connection. But we have to be able to do the work to advance the science and to understand the system. And so plant pathogens and the type of work that I described in that system are great ways to model the transport of domestic animal pathogens, human pathogens, and to do it under a fairly safe and secure way. So
It looked as though that farm was in an isolated area.
that's right. For the pollen stuff, absolutely. With the research farm, not so much, but we tend to have sort of borders. And then we are trapping the pathogen with drones and downwind, so it's moving. And so like I say, it's all about the connection and the communication. Great questions. That one actually comes up a lot. Yes.
Can you go back on the slides between Australia and New Zealand, and you were talking about the air bridge?
Yes.
And I was wondering, is there a correlation with, I don't know if there's like migrations of birds that go between New Zealand and Australia? And do planes use those air bridges? Do we have air bridges from Europe to the Caribbean? And I have another question, but I'm going to stay.
Yeah, so these are all great questions. So I'll try to summarize them if I can. She had a lot of questions about the air bridges. Are other things moving along with them? Are migratory birds using the air bridges? Are aircraft taking advantage of the air bridges to move more efficiently? The shorter answer is I don't know, but I'm going to ask Ilse because that's a great question. But we do know the insects move along these air bridges. And one of the things that Tony Withers has shared, she shared some of these incredible, she was the entomologist right here. She's witnessed these major migration events where all of these insects basically go up at once. They're responding to some sort of a swarm trigger. We don't know what it is. And then off they go across the ocean. Some of them she was sharing, some of them may travel fairly close to the water surface. Some may go up a lot higher. Depends on if there's rain. Of course, if they're flying close to the water surface, fish are jumping up and eating them. Birds are picking them off as food. So how these insects kind of know where to go, how to take advantage of a certain layer of the atmosphere, a certain bridge, we're only just beginning to understand. This concept is entirely new. We just published this paper describing this phenomenon just a couple years ago. So we're only just beginning to understand these air bridges, and it's a very exciting area of research for us. Could this help us think about more efficient air travel? Probably. Are there bridges connecting Saba to other islands, to other countries? Likely. But we haven't done any of that modeling just yet. Great questions. Did you have a follow-up?
Yeah, well, I'm just thinking about the presentation we had about the seaweed a few days ago or a couple
Sargassum,
of days ago. Yeah, and they're talking about basically it's going from pretty much the coast of Africa all the way to, passing the islands. Those are going to be things that you're going to need to look into as well, because there's going to be some other pathogens that are going to be traveling with the seaweed.
With the seaweed.
I guess.
Yeah, so the comment was, so I didn't hear that presentation, but it was a comment about sargassum and long-distance transport in the ocean and what could be moving with those. I don't know. That's a very interesting system and extremely problematic for coastlines, including here in Saba, right, when it shows up. I think I'd like to take that question and just bring it back to this idea of coupling these models with the atmosphere, the ocean, and land masses. And when we start to do that, we can see if there's correlations in the movement of the atmosphere, these ocean currents, these things like sargassum to be able to predict when they're going to show up. We need more connections. Yeah, I'm just not an expert in that space, but great questions. Okay, yes.
Coming back to insects dispersed atmospherically.
Yes?
Has anybody done any research on migratory birds? Does that come into their system when they're traveling across water, that they are going to hopefully find one of these insect
Plumes, yeah. Okay, so another bird sort of question. I'm not an ornithologist by any means, although I was able to spot the chicken at the top of the mountain on the trail. What I will share with you is there was this really interesting paper published in a top-tier journal, PNAS, many years ago that was foundational for some of the work that Shane Ross and I actually did together to develop some of our long-distance transport models. And what they did is they were able to determine these structures in the ocean upon which prey fish were basically following. And because essentially the food for the fish was clustering along these, I don't want to call them bridges, but corridors in the ocean. And these birds that were feeding out at sea, because they were following the prey, which was following the food, were actually following the corridors as well. So I don't think they were doing it intentionally, but there was a system that was governing the transport of the food for the fish, which was then governing the transport of the birds. So this connectedness across many different domains, I think, is in an interplay with your question. Yeah, so I mean, if you're a bird and then all of a sudden, you got thousands of moths and they're taking off, where are you going to go if you're hungry? Right? Yeah, or if you're flying up there and all of a sudden, a big swarm is coming, and you're like, hey, I could eat along my way, I guess off you go. But, I'm not a bird and I'm not an ornithologist. But these are all great things. I'm glad you're thinking about them.
So if we can call it there a little bit, I'm sure David would be very happy to have dinner with you or just answer more questions for you. Can you give us just 30 to 60 seconds of why we should join your field project on Saturday morning? Because I think a lot of us on Saba are very aware of the Sahara dust or concerned about pathogens. So tell us what you could explain to us on Saturday morning.
Yeah, so I've got a portable air sampler that I brought with me that has these really cool cartridges that we put into the sampler. I brought a number of cartridges, so for those of you that want to come out, you can actually run the sampler. And then I have this digital microscope, and we're going to look at what we collect from the air on the little cartridge. And so we'll run. I'm not sure exactly where we're going, but we're going to sample different locations. We talked about the museum and many other spots, depending on the attendance of folks and any mobility challenges that we have. We'll be accommodating. And so come be an aerobiologist for a couple hours, and let's see what's in the air. If you have any unique ideas about where you want to stick the air sampler, we can put one in the bathroom, by the cat litter box, whatever you want to do. We can be creative with where we put it. It's small. You'll see it. And it makes a nice little delicate humming noise. And it's just a neat way for people to appreciate what's in the air and what you're breathing on an everyday basis. And so please come out and take a look. I'm excited about the opportunity to work with everyone. And yeah, I'll let you push the little start and stop on the device too. So
Fabulous. So know that on Saturday morning, we're going to start at the museum grounds so we can accommodate everybody. You don't have to do a super hike. And then we're going to do a little bit more of a real hike after that, but you can get the comparison from both experiences. So thank you very much. Thank you, everybody. We will see everyone Saturday at Calibre, and it's not going to rain again as we wind up our couple last activities. So thank you for coming out, and let's keep it going. Thank you so much, David.
I do have one request. I'm going to do a little video selfie. If you can, we'll do 3, 2, 1. Everybody say aerobiology all at the same time. How about that? All right, 3, 2, 1. Aerobiology. That was special. Thank you for that moment.
By all means, make sure you pay your bill and join the Hideaway as our great sponsor and have a great dinner. Thank you, everybody.